Welcome back. This is the third part and the last part of lecture 7. We're going to go into chapter 19, the microbial disease of the skin and wounds. Okay, so the structure of your skin. Again, you have two main layers. This is something that you should already know. Your dermis and epidermis. And this is a sketch of it, a diagram. And the protective outer layer, epidermis, it's only invaded deeply in case you have a wound, and that's also known as the trauma. It could be cuts, scrapes, etc. And this is how microbes are allowed to go into deeper tissues of the body. Tell me why. Think of pH and think about salinity. Okay, the skin microbiota. What is important here, your normal microbiota will compete with potential pathogens, and that's what makes it very difficult for the pathogens to actually enter into your skin or below your skin. Some of the microbes that you find of the normal microbiota, the yeast, okay, and you find also some of the bacteria which are known as the staph, micrococcus, and the propionobacteria, many more, okay. They may produce disease if they do penetrate the epidermis, or again, if your immune system is suppressed. Tell me why. And this has to do with your normal microbiota too, okay? Of course. Okay, so let's talk about bacterial disease. Folliculitis. That's where your hair follicle basically grows into the skin instead of outer skin, and that causes the pimple. Okay, it also can cause a sty when it occurs in the eyelids. It can spread into surrounding tissues, and it produces a furuncle. Carbuncles occur when multiple furuncles grow together. Most are caused by the Staphylococcus, and these are gram-positive anaerobic bacteria, facultative anaerobic, that is. They're cocci, and they're also in your skin because they happen to be tolerant to the salt concentration of your skin. You have Staph epidermis and Staph aureus. And you're familiar with it. You saw this in your microbiology lab. We do have ways to identify what type of staph. We have it is a positive catalase that will make it a staph, and then you can do a coagulase test to actually see which ones are positive versus negative, and that will give you different species. And lastly, we can also do a hemolysis test, and that's what you see here on the actual area when we talk about hemolysis. So I'll give you this answer. What is it called? This table gives you comparisons of the two staph species, how we can identify them. Now think about why can you take or can you not take penicillin if your beta-lactamase present in the actual organism. Can you take penicillin or not? Moving on with folliculitis. Well, again, the staph is actually transmitted via direct contact or by fomites. Okay, fomite is another object. The infection is present to your blood. It can move to organs if it's become systematic. We can do the epidemiology by looking at what type of staph is it. Okay, the staph epidermis likes the virulence, so it really causes disease. Some of the disease caused by Staph aureus. Okay, the diagnosis, do a gram stain. Gram-positive bacteria is the first thing that we have to deal with, okay? And then we can do a coagulase test, and it can be treated with dextroglycan. Now, this works and not penicillin, because this happens to be a semi-synthetic penicillin, which is resistant to the beta-lactamase. So that's the answer that you probably gave me before. Vancomycin is also used to treat the resistant strains. And wash your hands. That's how you prevent it. Proper use in hospitals to minimize MRSA is also important. And as you know, MRSA is a type of methicillin-resistant Staph aureus. Okay, the Staphylococcus scalded skin syndrome. Yep, not very good looking, as you can see. Signs, well, it becomes very red. The skin becomes very red and wrinkled, and the outer layer of your tissue, the epidermis, peels off in sheets, as you saw in the picture. The pathogen is some of the staph strains, and what happens is that they release some of the toxins that breaks down your desmosomes, and that is the cell-to-cell junctions in your epidermis. There is no scarring because the dermis is unaffected, so basically your skin will grow back, and the death is very rare. It occurs mostly in infants because their immune system is not as highly developed, and their skin tissue is also not as strong as the adults. It is transmitted person to person. It's easy to diagnose, you know, as you saw the picture that's out in the skin. You can treat with topical drugs, and the prevention is difficult because your Staph aureus is in the skin. Impetigo, okay, that's pyoderma, and you also have erysipelas. These are also diseases of the skin. You get impetigo, you get these red patches form in the face and limbs. And the erysipelas, you have infection spread through the lymph nodes. So this is your impetigo right here, as you can see. And this is your erysipelas, it goes into your lymph nodes. Again, Staph aureus is the most time the virulence factor. However, sometimes the Streptococcus pyogenes also causes this. This is gram-positive coccus, just a staph. And what happens, they have these proteins which are known as M proteins, and they actually attack the factor H, and therefore it doesn't let us, it doesn't let the immune system attack it back effectively. They have this enzyme also known as hyaluronidase, which breaks down the hyaluronic acid, and this acid is necessary for connective tissue and immune system to work properly. And they also release these toxins, which causes rash and toxic shock syndrome. Okay, in the pathogenesis component, basically the bacteria invade when the skin is compromised, and but most important, it can actually cause acute glomerulonephritis, which means it's going to affect the kidneys. Very damage to the kidneys. It could be very dramatic. It can be transmitted person to person, you know, via contact or fomites. However, impetigo is mostly in children, but the erysipelas is also more likely in the elderly. How you diagnose? Well, just by looking at the actual skin, you can see vesicles. Okay, it is treated in the case of impetigo with topical antimicrobial creams and constant cleaning of the area. In the case of erysipelas, it will be treated with penicillin. It is prevented with good hygiene and clean hands, and always good hygiene is the most important thing. The next bacterial disease is the necrotizing fasciitis. It's caused by strep pyogenes, which are your group A streptococcus, and they have various enzymes that actually help get through your tissue, especially through your epidermis, that is. They release exotoxins A, that means on the upper surface, that's why it's called exotoxin, And they also have streptolysin, which would cause damage to your cells and tissues. Your pathogenesis, well, how does it happen? Because of the streptolysin, your strep pyogenes enters through the skin by breaking it, and it can be spread through person to person. It is diagnosed, well, it's actually difficult because the symptoms are not that easy to diagnose. You start feeling like you're under the weather, like you have some kind of common cold, but then, boom, it hits you really hard. It is treated with different types of antibiotics, as you can see, clindamycin and penicillin. And here you can see the pathogenesis. The streptolysin is the one that causes the skin to break like that, and then the empathy can bind to the surface. The ectotoxin triggers the overactive immune response. Good old acne. Well, most people think acne is only in adults, but it's not true. It can happen at any age, but it is more commonly early adolescent age. It is most commonly caused by a bacteria known as Proteobacterium. It is a gram-positive. It is a rod-shaped diphtheroid. It looks like the picture you see right here. It is a Corynebacterium. However, it's not pathogenic. It is found in your skin, and it loves the oily areas. That's why we say be careful of the oily teenagers. Okay, as you can see, from stages 1 through 4, once the pustule has finished, that's when you get the pus, and it goes out through the skin as the hair follicle is coming out. Your propionobacteria happen to be normal microbiota. Okay, like I mentioned earlier, it happens more in adults, so it can occur later on in life. How you diagnose this? It is through visual examination. We can treat it with microbial drugs, antimicrobial drugs, and these are the ones that can cause exfoliation of the dead skin. Accutane is mostly severe, used for severe acne. However, UV is also used to destroy bacteria, but obviously we're not going to use UV in the face of adolescents. Okay, we'll continue with bacterial disease. The next one is called the cat scratch disease. The signs are first thing, of course, the fever, malaise, and you have a localized swelling in the infection site. And you can see right here that it causes your lymphadenopathy, meaning the lymph nodes can actually increase in size. It is caused by Bartonella henselae, which is a gram-negative bacilli. As you can see there on the picture, that is a gram-negative. Okay, and the endotoxin is the one that causes this main virulence factor, the endotoxin released by the gram-negative bacteria. It's transmitted by cat bites or scratches and by blood-sucking arthropods. So that doesn't mean that it's only caused by a cat, obviously. How you diagnose? Well, serological tests like ELISA, and you can treat it with antimicrobial drugs. Not common infections. It happened mostly in Pseudomonas infection in burn victims. It's, as you know, it's a gram-negative bacilli. It's mostly found in soil. They can't matter in most environments. However, once it happens, it could be very deadly. Again, what happens? The bacteria kills the cells, it destroys the tissue, and triggers the shock syndrome, that is. But since these are burn victims, topical antimicrobials are the first sign to help them. The main problem is that once it enters your connective tissue, it can infect your organ systems. The way to find out if it is present or not is because the Pseudomonas releases a toxin known as phycocyanin, and this actually causes the discoloration in the massive infection that looks like a green color. This treatment is difficult, so therefore there is multi-drug resistance to Pseudomonas. This is not typical in healthy individuals. It's more likely, like I said, in burn victims. The next one we're going to talk about is Rickettsia rickettsii. This causes rickettsiosis, which is known as spotted fever. The tick is the one that causes this, with the vector, right? And it's also known as the Rocky Mountain spotted fever, which is the most severe of all these type of spotted rickettsiosis. Your signs at first, you get a non-itchy spot, and then you have a rash in the trunk and appendages of the victims. The most difficult here is that organ failure can happen. This rickettsii is gram-negative and is an obligate intracellular parasite. Therefore, it cannot live outside of its own cells that give them hostage. Rickettsia does not secrete any toxins, but it does have a major damage to your blood vessels, and it is again transmitted by a bite of the infected tick. You can find out if it is spotted fever through a serological test. We treat it with microbials, antimicrobial drugs, that is, and again, just like with other tick vectors that have to be avoided with tick repellent. And these are the number of cases of Rocky Mountain spotted fever in a decade of 2002 to 2012. Anthrax, big problem in the 90s, used for biological welfare. And what is anthrax? Well, it's a bacterial known as Bacillus anthracis, gram-positive bacilli, and it's an endospore forming bacteria. And it's hard to kill, okay, because of the endospore formation. It also releases toxins, and these toxins are the ones that contains 3 proteins, and which actually are the defensive factors for the bacteria. Because these are, when you have a patient that has been attacked with anthrax, it causes eschar, which is this black painless ulcers. It looks like dead tissue, as you can see in the picture right here of this patient. Requires controlled disease in animals, especially. You can get it from other animals. Okay, gas gangrene. This is caused because Eucrostidian species have the ability, which also endospore forming, they actually have the ability to survive very harsh conditions, and they secrete 11 toxins, which actually causes this gas to be created under the skin of the patient. The mortality rate, as you can see, could be 40%. The appearance of this gas gangrene is a given. And of course, rapid recruitment is immediate. If it's not removing surgical of this dead tissue, it will spread very quickly. Tell me why. Okay, now we're going to start with the viral disease of the skin and wounds. Many of these viral diseases happen to be systemic. Okay, so you know what that means, systemic. It can spread throughout the body. Okay, they manifest as signs and symptoms in the skin. However, there are many that go through your respiratory, meaning lungs, route first. We have different families of virus that causes viral disease. You have your pox viruses, herpes viridis. You have your paramyxovirus and your rubella virus. Okay, these are the family of the human viruses. When we refer to strand type, we're talking about the DNA, double DNAs or single-stranded DNAs or RNAs. But here we're looking at all DNA viruses, okay? So your double-stranded or single-stranded DNAs. Go. These are human viruses again. Continue, but now we're looking at RNA viruses. They call these single positives strands. These are more your GI and your livers. Then you have the other ones that can be positive or single strands that are negatives. These happen to be more your respiratory or happen to be systemic. Pox virus. Pox virus is a DNA virus, and it happens to cause the smallpox. And they also can cause other ones like the orf or the cowpox and the monkeypox, which is very rare in humans. This is the first human disease to be eradicated, the smallpox, and the signs and symptoms, well, it goes through these different stages. It goes into skin, little spots, and then it just goes throughout the whole body because of the actual toxemia or hemorrhagic fever that can persist afterwards. Okay, so as you can see, it goes into pustule, which is the pus, number 4. Then once it comes out, that's why they say when you have this, avoid touching it because then you develop too much scar tissue in your skin. The pox virus is happening again, DNA viruses. And the one that is known to cause variola is also pox virus. How do you get the smallpox or the variola? Well, through inhalation. They go through your respiratory tract and then through your body. However, the other pox viruses are spread by contact in your skin, contact to contact. Okay, although it is eradicated, we still have variola virus stocks that are maintained in the US and Russia for research. However, I must admit that it's not for good biomedical research. It's more for biowarfare. Monkeypox, it's another type of smallpox, but it's milder. It is spread amongst primates and rats. However, it has increased over the past decade. Treatment, immediate vaccination. However, vaccination discontinued in the 1980s because the smallpox was eradicated. Chickenpox and shingles. Chickenpox are highly contagious. They're characterized by the lesions on the back and trunk, as you can see in the pictures. And then after you actually get out of it, it becomes latent, but it becomes latent within your sensory nerves. And that's what causes shingles later on in life, okay? Is when the actual chickenpox virus becomes active, and then it actually leads to shingles, which are lesions localized in the skin. The varicella virus is the one that causes your chickenpox, and it causes both disease, by the way. And we have to understand that the reactivated varicella virus causes shingles, and the person that has shingles can also give chickenpox to somebody who never had it before. It is mostly, chickenpox is of course mostly in children, but nowadays it's not. I mean, it's not common at all because most children now get vaccination before they get it. Okay, another viral disease of the skin and wounds would be herpes infection. The most common that you know or you heard about is probably the herpes virus 1 versus herpes virus 2. The pathogenesis is basically you have painful lesions caused by inflammation and cell death. And that's the one that you see right here, for example. The one in the mouth that you can see here, it's mostly caused by herpes 1. However, herpes virus 2 can also cause it, and herpes virus 1 can also cause not only lesions around the mouth area, but also in the genitalia now. How does it spread? Well, it spreads through mucous membranes of the mouth and genitals. They're not life-threatening, but it could be very different for neonatal. They can get into internal organ or central nervous system of the neonatals, and that could be very fatal. It is made the presence of the lesions. Once you see it, you know if you have herpes 1 or herpes 2. There is an immunoassay that reveals the presence of the amount of herpes type that you have, or the patient may have rather. There is drugs that help control the disease, but it doesn't mean there is a cure for it. And that is the drug right there, acyclovir. Warts, HPV. Okay, HPV. It's the one that also the another one is the papillomavirus. There's different types of HPV that can cause this. As you can see, there's so many strains that some of them can even lead to cancer. Okay, how do you know if you have warts? Well, they actually develop several months after the infections. Okay, they're harmless, most of them, but they can precipitate into cancer later on in life. How do you get it? Direct contact. That's how you get it, via direct contact. You can get it from skin to skin or through an STD. The cancers can be treated, and there's actually vaccines that are given now to adolescents, especially young ladies, because the HPV can cause later on cancer in the female reproductive system. And they're given in different amounts through different 3 to 5 vaccines. It depends on the ones that we actually are looking into. How do you remove warts? Well, there's different ways that dermatologists can do that. Okay, now we're looking at comparison of measles versus rubella. Okay, the rubella virus happens to be an RNA positive virus, and that's the way they actually use the antigen positive, antigen negative to actually replicate. And your measles, which is also known as rubeola, okay, rubella is known as your German measles, and measles is known as the rubeola, is an RNA negative strain. Your rubella is your German measles. Children develop a smell rash, but in the case of adults, it can develop into arthritis and even encephalitis. In the case of congenital infection, it can result in birth defects for the newborns. And the rubella virus, again, is caused by a single-stranded, positive RNA and a messenger RNA. It goes to the respiratory tract, and then what happens is the immune system contribute to the severity in the adults, because if you have a low immune system, it doesn't respond quick enough. The rubella is spread by respiratory secretion. It happens only for humans. Your diagnosis is simple. Look at the rash, and then we'll do a serological test. There are vaccines still available. However, for some reason, there was a high rate of cases in 2008, and you will know that very soon. It happened again in 2014. Rubeola, measles. It is characterized by complex spots. What is that? Well, those are it right there. When the patients open their mouth and they do analysis, you can see those complex spots right in the mouth. Okay, And you can also see the measles rash in the person's skin. The complications can be about 30%, and those can even go up to such things as inflammation of the brain, encephalitis, right, and pneumonia. You do have cases in which you get a sclerosing panencephalitis, but it's very rare. Okay. When you actually look at the pathogen, this happens to be an envelope virus, and that makes it more difficult. They also happen to be single-stranded RNA, and that means they're complementary to RNA. In order to replicate, that makes it very easy for them. They can have an adhesion fusion to proteins because they have receptors that can actually help them get into the host very easily. The immune response to infected cells is quickly. However, it can be fatal in children. Is it contagious? Yes, it is. Very contagious. Okay. However, the vaccination has reduced spread of the disease. Children are given the rubella vaccination earlier in age. Diagnosis, as again, you look at the signs in the skin and the mouth. And how do we treat it? Well, antibodies, ribavirin, vitamin A, and we do have vaccines that protect against it. MMR vaccine. And that's something that you need to think about. Why do we have such a rise in 2014 and it dropped again in 2015? Other viral rashes, the erythema infectionum. This is also known as the fit disease. As you can see, this young child with these nice pink cheeks, it doesn't happen to be something so nice. It's caused by B19 virus. It is a respiratory disease that actually goes into from the respiratory system into your dermis, and it becomes a rash. In the case of adults, it develops anemia and joint. You must have heard also about the variola virus. It happens to be herpes virus 6. Tell me why. And that has to do with the graph that I showed you earlier. Mycosis of the hair, nails, and skin. We're going to get into the fungi now. These are mostly opportunistic. And I surely hope you know what that means by now, opportunistic, okay? Some of the mycoses are identified or classified depending on what they actually do the infection. Some will happen to be, on the superficial, outer surface of your skin. And some of them just happen to be cutaneous because they go all over your skin. Subcutaneous is under, and systemic is, as you know, numerous systems. Some of the common of the thermophytosis, that means those that causes disease, athlete's foot, chalk H, ringworm. Become familiar with these. Okay, let's start with the superficial mycosis. These are the most common. Okay, that's when you get like fungus on your hair, nails, and outer skin. Okay, one of the symptoms of the pityriasis versicolor. Okay, and that means right here, as you can see, the skin, it has all these patches that you see different colors. That's why they call it pityriasis versicolor. Okay. This is caused by the malarious furfur. It is a normal inhabitant of the human skin, but which becomes actually opportunistic. So, what is the pathogenesis of the superficial mycosis? These produce keratinase. So these are enzymes which happens to break down keratin. So that means it can actually break down keratin in your skin and even in your hair. Okay. So how do you get it? Well, by sharing hairbrushes and example of combs. This is the ones that affect your hair. It occurs mostly in adolescent. Uh, I guess that's because adolescents share so much of the combs and the brushes is because the immune system is not as developed as adults. What is the diagnosis? The infected skin. It becomes pale green when you look at it under UV light. Okay. However, further diagnosis by microscopic examination and how is it treated with topical, mostly topical drugs and sometimes oral drugs. Okay, when we go into cutaneous mycosis, we have the dermatophytosis. Okay, look at the word dermis. These happen to cause cutaneous lesions, and sometimes by fungus that don't grow in the skin. These are dermatophytes. They happen to eat the keratin, and as we know, keratin is the protein in the skin. There are cell-mediated immune responses that damage deeper tissues, excuse me, and that means they can go even deeper into your tissues and cause more damage to your organs. This is your example of dermatophytosis, also known as the ringworm. Another example is your athlete's foot. Yep. Not very good looking. So what are the pathogens? You have three types of genera, okay? You have your microsporum, your trichophytins, and your epidermophotines flocosums. The hermaphrodites have, well, they colonize skin, the nails and the hair. And how do they survive? They use protein known as keratin to survive as a nutrient source. Infection is rare, but they happen to invade living layers of the skin. They are classified by natural habitat. They are anthropophilic. It means they're associated with human. If they're zoophilic, they are associated with animals. And if they are geophilic, they happen to grow in soil. What is the diagnosis and treatment? Well, clinical observation first, right? Most of the time, there's potassium hydroxide preparation of skin or nail samples to confirm that there is what type of infection or what type of mycosis. It is sometimes used with topical agents, but very unlikely because it is widespread. Then they use oral drugs, which, by the way, they are very damaging to the liver. The oral drugs. Some of them grow deep in tissues, but they actually do not become systemic. Okay, so they stay in the same site. They grow into the epidermis, but produce skin lesions eventually. Chromoblastomycosis, it is caused by 4 different species of the ascomocytes. Okay, and remember what are the ascomocytes in humans? They actually become golden brown, and so they call them golden brown bodies in the skin. That's how you see the diagnosis. You have to remove this infected tissue and then give antifungal drugs, to the patient. This happens mostly to people that are working barefoot in the soil. So therefore, how do you avoid it? Well, wearing shoes that reduces the steps or number of infections. As you can see, it's not a very pretty sight, not at all. The phytohyphomycosis, it's caused by more than 30 types of fungi, and it's most likely when you have a spore that can enter the wound. And again, that happens with people that work, close to the soil. And how do you know what type it is of the fungus? Well, we look at the hyphae in the skin sample, and then we also do a biopsy, and they can also do a CSF analysis. The mycetomas, again, soil fungus. They actually can cause tumor-like lesions. And this happens to be more prevalent in areas near the equator because of the actual humidity in the soil. And they you have to treat it, you have to actually do surgery and then continue with antifungal therapy. Okay, the sporotrichosis. These also, the sporotrichs, they actually also reside in the soil. Again, people that work in the farms mostly get it. They call this rose gardener's disease. It causes mostly in the, it's more limited to the arms and the legs, okay? And they remain local. These fixed fungus, they don't go systemic. However, it can attack your lymphatic system, and that is known as your lymphocutaneous sporotrichosis. And this is a picture of it. Wound mycosis. These are caused, again, sporotrichosis. How is it diagnosed? We look at the patient's history, and most important, you have to look at the type of fungus that you have in the clinical samples. It causes lesions, which can be treated with antifungal drugs. Again, prevention, wearing the proper attire. Tell me why. Parasitic infection of the skin, leishmaniasis, produces large painless skin lesions. These skin lesions enlarge to encompass mucous membranes when it becomes mucocutaneous. When it's visceral, it means it can go throughout your body, through your viscera, means the other organs. Leishmania is causes an alien, but this is not now a fungus. This is a protozoan, okay, which is transmitted by female sandflies. So the infected macrophages, once they actually see this protozoan, they stimulate the inflammatory response. This happened to be only in parts of the tropics, subtropics. Diagnosis is microscopic of the protozoan. Most cases, believe it or not, heal without treatment. However, antimicrobials are needed for severe infections, as depicted you saw previously. And the exposure to reservoir hosts is, to the sandfly that is the best way not to get this. Okay, so we move on to scabies. These are your mites, correct? They're known as Sarcoptes scabies. And basically, they burrow under your skin. And once they burrow under your skin, they lay down their eggs. And each nest that the people get is because when their eggs are actually open up, they actually goes out through the skin, the new larvae, and that's what causes this crazy itchiness of these mites coming out. How you diagnose? Well, you can observe the mites, you can look at the eggs, you can even get it from the fecal matter of skin or even the skin samples. How you treat it? Mite killing lotion, and make sure you boil those items that have contact to it. I'm talking about personal items that you have, like for example, bed sheets and all that. Hot water. And that will conclude the third part of lecture 7. Thank you. Have a good day.